What's up, YouTube? Hopefully you're ready for this. This is going to be a fun lecture. I can't wait. It's going to be a longer lecture. I'm going to warn you right now. I'm not sure how long because I'm going to make it, but this is going to be a fun lecture. And what we're going to talk about are the receptor basics. Now, I already have a receptor video, but I really wanted to add to it. I really wanted to be more complete, and I think this video should help do that. So I kind of wrote an outline both for myself and for you. Just kind of cover stuff that I want to talk about. First, we're going to start with the basic receptor categories. We're going to do like a little flow chart where like we break it down kind of like this and maybe more like that, kind of like a little flow chart. Next, we're going to do the body breakdown where we actually see these receptors in the body. That's always a fun one. And then next we'll talk about what G receptors are. So we'll talk about what G receptors are and the different types of them. And then we'll talk about the different types, and that's where it kind of gets boring. But then we finish up with the functions of the postsynaptic receptors. That should be okay too. Yeah, you know, this is gonna go well. So let's just go ahead and dive in here. So I already promised we're gonna start with kind of like a little flow chart of what we're talking about. So let's start up here in the middle. We've got receptors. I gotta warn you, my spelling's pretty bad, so if I'm spelling receptors wrong, don't yell at me, but that looks right to me, so I'll leave it at that. There are going to be two main types of receptors in the body. Yes, there are more. However, for my sake, there's going to be two main types. There's going to be cholinergic, and there's going to be adrenergic. You may have heard these before. What do they actually mean? Well, cholinergic's going to deal with acetylcholine, ACH. Acetylcholine is going to be a neurotransmitter. I have an excellent video on how acetylcholine is synthesized. Botulism toxin deals with acetylcholine, but that doesn't matter because we're just talking about cholinergic versus adrenergic. Then adrenergic is going to be broken down into some different types. We've got alpha adrenergic. We've got beta adrenergic. We've got some others. We've even got like dopamine receptors. We've got histamine, etc. That's beyond the scope right now. So this is where, if you hear what an alpha receptor is, it's just a simple type of adrenergic receptor. If a beta receptor is just an adrenergic receptor. However, cholinergic receptors are different. These are going to be more like your nicotinic, and then also muscarinic. No, nicotinic not standing for nicotine like what's in cigarettes. No, nicotinic is going to be an ion channel. So it's going to be a sodium-potassium channel. Ion channel. So this one's going to be a little unique. I'm going to kind of squiggly it because this is going to be unique. And why do I say it's unique? Is because it's not going to use G receptors. And like I said in my outline, we're going to have some fun times and some not so fun times with G receptors. So we have a nicotinic receptor. So nicotinic is going to deal with the neurotransmitter acetylcholine, while adrenergic is going to deal with like norepinephrine or epinephrine. Here in the case of dopamine, we'll be dealing with dopamine, etc. So cholinergic deals with acetylcholine. There's two different subtypes. The nicotinic subtypes are going to just have an ion channel. The receptor is actually a channel, so when this acetylcholine binds, this channel will open up and stuff can go into the cells, like sodium can go into the cell. Vada vi, vada vu. I don't even know if that's a word, but we're going to go with it. So notice how the nicotinic don't have no G proteins. That is a take-home message. No G proteins are associated with the nicotinic cholinergics. However, muscarinics, they're, yes, G proteins. And then same with over here. All this stuff over here is going to be, yes, G proteins. Okay, good. So we're talking about G proteins, and we'll cover what that means. Just for now, know that. There are two different types of nicotinics. There's going to be a neuromuscular, or an N and an NM. An N is going to be for autonomics. Here, I'll break this down even a little better for you guys. so maybe you'll be able to follow along a little better. We've got NN and NM. So the NN is going to deal with our autonomic function. Now, where have you heard the word autonomic before? Fight or flight, maybe rest and digest. Autonomic, so like the sympathetic and the parasympathetic system. Well, NM is going to, the M I like to think for muscle. So it's going to deal with skeletal muscle. Skeletal muscle. So skeletal muscle, if I want to move my bicep, if I want to curl my bicep, I'm going to have my brain tell my muscle, and what it's doing is it's going to release acetylcholine, and that nerve is going to release acetylcholine, and that's going to act on my skeletal muscle at a receptor that's going to be a nicotinic subtype M receptor. However, let's say that I wanted to activate a sweat gland, for example, or anything else, my preganglionic, and I'll get to that. Don't worry, don't be shaken. Preganglionic versus presynaptic versus postsynaptic, or preganglionic versus postganglionic. I'll be talking about that, but right now, just know that the preganglionic or the presynaptic nerves will spew out some nicotinic, or will have nicotinic receptors. So let's actually go to that now. So this is the basics. Just realize G proteins are on pretty much everything except for this nicotinic receptor. The nicotinic receptor, so when I say receptor, I guess I should have done this. We have got a nerve terminal here, and then we've got our, it's either another nerve or it's going to be an organ. So I'm going to write organ or nerve. And this is our nerve or target organ or something, just whatever we want. We're going to secrete, let's say ACH. This picture is looking a little dirty, so I'm going to get rid of that the more I think about it. We've got some ACH here in the, doesn't look as dirty. So we've got some acetylcholine released from our axon. It's going to find a receptor here, and depending on what kind of receptor, are we talking about a nicotinic receptor, are we talking about muscarinic, we're talking about any of these adrenergics, and that'll mediate the function. So the receptor type really kind of determines what will actually happen next. So that's what we're talking about with receptors. Okay, so let's move on here. We've got the body. Hold on, wait for it. Here we go, body breakdown. We're going to have some fun with the body now. Now we've got a little chart here. You may have seen this before. We've got our brain. It's up in our system. Next we've got our spinal cord. Our spinal cord is going to extend down to like L3-ish, and it'll form the cauda equina. You'll see the cauda equina down there. Okay, so just orienting you what this is, since my pictures lack in detail a little bit. But we've got cauda equina drawn in. Alright, so we've got the brain. Right below the brain you've got a medulla and like your brain stem, and that's where a lot of your cranial, you'll have some cranial nerves come out. So I'm just going to write in a different, probably I'll keep green for a little bit longer. Just remember, this is the cranial. I warned you about spelling. This is the cranial. Okay. We've got the cranial, so we're just below the brain. We're in the brain stem. Now we've got nerves coming out here, and this is going to be our parasympathetic nerves. So if you've heard before, parasympathetic are going to be craniosacral, while our sympathetics are going to be, wait for it, thoracolumbar. If you already knew that, great. However, this is what it means. Our parasympathetic nerves will be coming from our cranial, so near the head, and the sacral, so near the sacrum region, while our sympathetics will have a sympathetic chain associated with it, and I'll cover that. But they'll come out near our thoracolumbar region, so our thoracic lumbar region. So we're up near the head right now. We're in the cranial region, so we're going to deal with the parasympathetics. Fun stuff. Now, when I do this, it means that it's going to synapse. So just like here, we're going to synapse. So here's our presynaptic neuron, and then if this was a nerve, not an organ, this would be our postsynaptic neuron. Our postsynaptic neuron is going to then go on and do other stuff. So that's what I meant. This is one nerve. It's going to synapse. You're going to have the synaptic cleft and everything. You're going to find some receptors on this nerve that I'm drawing in. So there will be receptors over here, and depending on that receptor, you'll get a different effect. Then it's going to synapse again. So that's what the little crocodile mouth looks like. One nerve is eating the other. So we've got a presynaptic neuron, meaning nerve. So presynaptic nerve. Then we've got a postsynaptic nerve. Postsynaptic. And then, so we've got the sympathetic chain or the sympathetic ganglion. This also can maybe be referred to as preganglionic depending on the circumstance. So the presynaptic nerve and the postsynaptic nerve. That's a good point to remember. Okay, so now let's go a little further. So we have receptors here. I already said we have receptors here. So what are they? They're going to be acetylcholine. Acetylcholine is going to be everywhere. It's going to be rampant. We have lots of acetylcholine. Now, I said there's two different types. There's nicotinic or there's muscarinic. Let's go back really quick because this is important. Oops. This is important. So I just said we have acetylcholine. Now, we got to specify what kind. Well, I'm going to tell you that it's nicotinic. Why? Because we're autonomics. Here's the autonomics. Remember, autonomic is going to come in. The parasympathetic, the fight or the flight, the rest and digest, all that garbage. That is the autonomic. So we're going to deal with a nicotinic subtype N specifically. But from here on, I think I'm just going to write N, not subtype M, subtype N. Told my spelling was pretty bad. That's because of this pen that I have. Acetylcholine NN. And that's going to be our autonomic. So the receptor type over here will be NN. What I have not circled is going to be the neurotransmitter released. So that's going to be acetylcholine. So we're going to have some acetylcholine release, attacks the NN receptor, and then it's going to mediate its effect to the postsynaptic neuron. That postsynaptic neuron is going to meet our parasympathetic innervations, whether it be muscles, whether it be glands, oops, not bands, glands. So the muscles, I mean, we're talking like heart muscles, it could be smooth muscles, or just like any gland in the body. I'll cover that later. Right now, we're just trying to learn. So then we've got a thing called the sympathetic chain, paravertebral chain, the paravertebral ganglion. It's going to be a chain, and so here's our spinal cord. This chain is going to run along the vertebral bodies, and this is going to indicate our thoracolumbar region. This is where our sympathetics come out. Parasympathetics don't come out in the thoracolumbar region. The parasympathetics will come out in the craniosacral region only. So anything in here that's autonomics, autonomic meaning parasympathetic or sympathetic, is going to be sympathetic because we've got our thoracolumbar region. So that's where we're at right now. We're in the thoraco, and then here would be the lumbar region. So there's going to be three main things that come out of here. Now we're going to synapse, meaning we're going to go from our preganglionic to our postganglionic, so the sympathetic chain ganglion. So we're going to have, these are be postganglionic nerves then, or postsynaptic nerves, meaning here's the preganglionic, because here's our sympathetic chain ganglion, preganglionic, postganglionic, presynaptic, postsynaptic, whatever you want to say. Always, 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 we are going to see acetylcholine released here. But wait, sympathetics, doesn't that deal with like norepinephrine and epinephrine? Yes, it does. But right here in our sympathetic chain, we are going to be using acetylcholine neurotransmitter, just like we were here. So it's going to be acetylcholine transmitter. Remember, acetylcholine transmitter. This is just from nerve to nerve. This isn't from like nerve to organ and then that organ to the rest of the body. No. Acetylcholine with a nicotinic subtype receptor within that sympathetic chain. All of these. So that's why I just wrote it once. Okay. So remember that. Next, we're going to talk about some different targets. So this will be a different target, this will be a different target, and this will be a different target. So we're going to start with what you typically associate, and then we'll talk about some of the oddballs out, because medicine is never easy. The body is always complicated. You can't just have an easy system, otherwise everybody would be doctors. So this is what's going to separate you from everybody else, is you're now going to know what this chart means. So we have acetylcholine and a nicotinic receptor in our sympathetics. Oh, I didn't go over here. My apologies. I just looked back. We've got acetylcholine. Except over here, we're going to have a muscarinic receptor. We're dealing with a nicotinic receptor right here, and we're going to deal with a muscarinic. Muscarinics have those G proteins. And then over here, what we're going to do is we're going to innervate some muscles again. We're going to innervate some glands. So kind of like we did over here, glands and muscles and organs and everything else in your body can have parasympathetic or sympathetic innervations. And sometimes they have both active at the same time, but for our sake, we're just going to have muscles and glands. We're going to have norepinephrine is going to be our main neurotransmitter. And you can have alpha, you can have beta, you can have alpha 1s, you can have beta 1s, you can have beta 2s, you can have a whole assortment. So I'm going to leave all that out. But for receptors, I'm just going to say you can have adrenergic receptors. Adrenergic receptors here. So we used acetylcholine from our nerve to nerve, and from our nerve to our target tissue, so our muscles and our glands, so from nerve to target tissue, we're going to use the norepinephrine. Here we have the acetylcholine to the nicotinic, and now we'll be innervating sweat glands. Sweat glands are a little different. Sweat glands are like the odd man out. They're going to be sympathetic. However, they'll be sympathetically innervated. So when you're being chased by a scary monster, you're going to be sweating. Your sympathetics will be in overdrive. You will sweat. So your sweat glands are sympathetically controlled, except you're not going to be using the norepinephrine, like norepinephrine and epinephrine. Instead, you'll be using acetylcholine, and that'll be a muscarinic. Well, why is that? You can have acetylcholine, and that's still sympathetic? Wait, I thought acetylcholine was mostly parasympathetic. Mostly it is. However, sweat glands are going to be a zebra. They're going to be one of those oddballs. That's why medicine is not easy. Okay, and then one more. I'll pick this up a little because this video still is going to be pretty long. We've got dopamine receptors. And where are we going to find these dopamine receptors? We'll find it in the kidney, smooth muscle. And I'll say what that does later on. Just remember that dopamine finds dopamine 1 receptors. Dopamine 1 receptors. Dopamine 2 receptors are pretty much brain stuff. Dopamine 1 receptors you're going to find in the kidney, smooth muscle vasculature, so blood vessels. I'm going to write the smooth muscle and the blood vessels. Got a couple more. Stick with me. Stick with me. We've got adrenal gland. The adrenal gland innervation. So I want to leave this in, adrenal gland. Okay, so we've got our little synapse there. Adrenal gland. Why doesn't this fall under the chain here? Well, it's because our adrenal gland is going to be directly innervated. Directly innervated. Our adrenal gland, if you can remember, especially in the medulla region, so you have the cortex of the adrenal, and you have the medulla of the adrenal. The cortex is going to synthesize like your aldosterones, etc. I have a video on that. It's a great video. However, your medulla is going to synthesize your catecholamines, which I have a video on. I have a video on a lot of stuff. It's because it all interconnects. So your adrenal gland, your adrenal medulla is going to synthesize your norepinephrine and epinephrine, and that's what you typically think of your sympathetics. If you have norepinephrine and epinephrine floating around in your system, you think sympathetics, and that's where that comes from. But notice how we don't have a presynaptic and a postsynaptic fiber. We just have a presynaptic fiber that's going to directly innervate the adrenal medulla, and that adrenal medulla kind of acts as this postsynaptic nerve to release a hormone. So that's another kind of like oddball out. There's a lot of oddballs out. I'm going to talk about another. So kind of, we're not talking about parasympathetics. We're just going to be talking about muscles. Let's erase that. I can write that better. Muscles. And we're not just talking about any muscles. We're talking about skeletal. So skeletal muscles. Skeletal muscles will be innervated by, wait for it, there it is, nicotinic receptors subtype M. Okay, remember, still cholinergic, still nicotinic. We're not talking about muscarinic, we're talking about nicotinic subtype M. While everything over here, so like this NN, those are all N subtype, big N subtype N. Over here, we'll be talking about big N subtype M receptors. M as in mom. Okay, we're still going to use acetylcholine here, and we'll directly innervate skeletal muscles. We don't have like a presynaptic and a postsynaptic. No, we just directly innervate it, kind of like the adrenal gland. So that is going to be our fun. Now, where do we see this? I drew it down here by the end of the spinal cord. You can see this at any level. Why is that? Because we have skeletal muscles at any level. So you're going to have different types of nerves coming out of your spinal cord. You're going to have motor. So I'm just going to say these are motor nerves. You can have autonomic nerves. Those are nerves that are going to be your parasympathetics or your sympathetics. So are you kind of understanding where I'm coming from? That's why I'm doing this, is I just want you to understand what we're dealing with, because the body is complex, but I'm here to try and make it a little less complex so you're mastering the concept of it. So now I'm going to go to our outline, see what we should do next. What's up, G? So let's talk about what G proteins are. There's 3 different types that we're going to be concerned about, and I'm just going to cover the basics. The basics. If you're a science master, I don't even know why you're watching this video, but I'm just going to cover the high yield, the basics of G proteins, because I could make a video on G proteins and it would be a long video. However, I'm not. I'm just going to talk about the basics. So we've got a membrane here. Remember, membranes are going to be phospholipid bilayer, so it's going to be a bilayer. We have the extracellular space. So extracellular space, meaning we're outside of the cell, and this will be inside. Okay, so we're gonna deal with 3 receptors, and I'm just gonna draw my receptors like this. G receptors are a lot bigger than this. They're gonna span the membrane more than this once. However, for our sake, we're just gonna deal with this. So we're going to deal with 3 types. There's 3 types of G receptors. G receptors. And we're going to go through their mechanisms. There's GI, GS, and GQ. We're going to make believe that this is a GS, this will be a GI, and this will be our GQ. And I separate them out for a reason. It's simply just because they need room. And it depends on what kind of cell. We're going to pretend this cell is going to express all 3 receptors. In real life, they're not probably going to express all 3 receptors. They may express a couple. However, just realize that these will be in different areas of the body, and then the functions will be later on. I have kind of a tie together at the very end that we can do for function. So I'm just going to show you what the mechanism of each G receptor does right here. So we've got 3 little subunits. So the receptor spans the membrane, and on the inside layer of the membrane, we've got 3 subcomponents. We've got a beta subcomponent, we've got a gamma subcomponent, and we've got an alpha subcomponent. Don't care about these two. Not sure what it did there. But we don't want to care about the beta and the gamma. We just want to deal with the alpha. Well, what's going to happen is the alpha is going to be in the form of a GDP. Kind of like ADP. Remember in ADP? ADP is the diphosphate, meaning there's only 2 phosphate bonds, versus ATP. ATP is going to have 3 high phosphate bonds. So we're going to convert this alpha unit, which is going to be GDP, into alpha GTP. We're going to add a phosphate. And what that phosphate is going to do is it's going to give it energy. Now, for the GS, it's going to, so the alpha unit will come over here. It will stimulate this thing. However, on the other side, we have GI. GI, S for stimulate, I for inhibit. So we're also going to have a beta, gamma, and an alpha. The alpha will be in GDP, just like this is. We're going to make it an alpha GTP. It'll migrate over. Remember, GI for inhibit. Inhibit this thing. So what is this thing? It's going to be adenylyl cyclase. Adenylyl cyclase. It's going to be an enzyme. And what this is going to do is it's going to take ATP, adenosine triphosphate, and convert it into cyclic AMP. Cyclic AMP. We're going to go camping. Adenylate cyclase is going to go camping. We're going to increase our cyclic AMP levels. Now, what is our cyclic AMP level going to do? So what this is going to do is we're going to increase protein kinase A. Protein kinase A. When you hear kinase, you want to think phosphorylation. And what this is going to do is it's going to phosphorylate stuff. Now, why do I say stuff? It's because that's as specific as we need to get. Once we have protein kinase A activated, we're going to phosphorylate other enzymes. Once enzymes are phosphorylated, they'll go off and do their thing. We're going to have a reaction. However, for our sake here, protein kinase A increases with camp activity, phosphorylate stuff, bada bing, bada boom. So that's the GI-GS. GS, you're going to have stimulating adenylyl cyclase. That's going to stimulate camp production. That's going to increase your protein kinase A. When you hear protein kinase, I don't care if it's type A, type B, type C, type Z. Think phosphorylate stuff. That's it. That's it. Basic. So now we've got a G protein, or a G receptor. My apology. We've got a G receptor, and we've got some alpha units, some beta units. So I'm going to write this in: alpha, beta, gamma. Again, we're just dealing with the A subunit, the alpha subunit. It's gonna be in the form of GDP. We're gonna add a phosphate, so now it's in the form of GTP. It can go off, do its thing again. So this alpha subunit dissociates from this. Same thing as over here. Same thing we saw. The alpha unit's gonna dissociate. It's gonna bind this thing. Same concept, except for GQ. GQ is a little different. It's going to act via a different mechanism. GS and GI act via the same mechanism. There we go. That's why I lumped them on the same side. So it should make a little sense of why I did that. Phospholipase C is going to be what this GQ ultimately stimulates. We're going to stimulate this phospholipase C. Now, stick with me here. It's going to convert PIP, which is going to be made from like lipids from this bilayer. And what it's going to do is it's convert it into IP3. And it's going to convert it into DAG. So the DAG is the diacyl glycerol. IP3 is a long word. It's inositol 1,4,5 triphosphate. Not important. Triphosphate, P3, 3 phosphates, triphosphate. Here's an inositol. It's at each one of these locations. Doesn't matter. It's an inositol, diacylglycerol. IP3 and DAG. Kind of has a ring to it. Now, each one of these is going to go off and do its own thing. So this is going to go and increase protein kinase C. Protein kinase C. You're saying to yourself, where have I heard that before? Protein kinase! Remember I said protein kinase is going to go off phosphorylate stuff. This is going to go off and phosphorylate stuff. IP3, on the other hand, is going to increase calcium release. Calcium is going to be stored in your smooth endoplasmic reticulum in most cases. It might be stored in other places, however, think smooth endoplasmic reticulum is going to release some of its calcium. Calcium inside the cell is going to do stuff. And what calcium is going to do is it's going to activate protein kinase. So it's going to activate a protein kinase that's going to be calcium sensitive. And this protein kinase will phosphorylate stuff. So what we're doing is G receptors take an outside molecule. So we've got a molecule of norepinephrine, let's say. We've got a molecule of norepinephrine. It's going to bind extracellularly to this G protein, or it's going to bind extracellularly to these G proteins, and it's going to initiate an alpha subunit to dissociate. Doesn't matter which alpha you're talking about, alpha units. Those alpha units, so an alpha unit that's specific to the GI receptor is going to inhibit. GS is going to stimulate adenylyl cyclase. GQ will do phospholipase C and convert downstream effects. The downstream effects, that's all this is. So hopefully you've kind of understood me so far. What we're doing is we're taking a molecule that's outside the cell, stimulating a receptor, and then ultimately we're phosphorylating stuff. And when we phosphorylate stuff, it'll be active. Okay, so now we can move on in our discussion here. Let's move back. G boring stuff. Yeah, we covered some G boring stuff, but we still have a little bit more to go, unfortunately. So let's do it. Buckle your seatbelts, because this is going to be one of those things where route memorization comes in. Now you're saying to yourself, "Oh, route memorization! I already memorized so much stuff." Well, think of it this way: it's route memorization, but I'll give you some good tools. I'll give you some good ways to remember this, so that way you don't have to remember as much. Okay, so there's going to be different types of receptors. You're going to have muscarinic. Remember, I said you have muscarinic receptors. You're going to have adrenergic. I'm just going to go back really quick, because G receptors. Remember, G receptors were found in a lot of places. G receptors were found in the muscarinic cholinergic receptors. So yes, we have muscarinics that are G alpha units. Yep, those are muscarinic, or these are G protein mediated. Betas, yep, G protein mediated. Dopamine, yep. Also histamine, etc. We're going to deal with a lot of G proteins. G proteins. What does that mean? Well, it means we're going to go via these mechanisms. While the nicotinic, I'm just going to draw down here, are nicotinic receptors. Those imagine as a channel. So we're going to have a molecule of acetylcholine come here. It's going to bind on the outside. What it's going to do is it's going to open up this channel. And what we'll have is, let me see here, wait for it. We're going to open up this channel here. And stuff is going to go through it. And then it'll eventually close, and all's good. So that's going to be our non-G protein. Stuff will just go directly through. Okay, now let's go onto the different types. So we have muscarinics. You've got three different muscarinics. You've got M1, M2, and M3. You also have more. M1, 2, and 3 are going to be your main ones. Remember, I just said I'm going to do the basics. I'm not here to teach you everything. I don't get paid enough to teach you everything. I don't get paid anything to teach you everything. So I'm just going to cover the basics. Adrenergic receptors. So you're going to have different adrenergic receptors. You're going to have alphas. You're going to have alpha 1, alpha 2, beta 1, beta 2. You're going to have some dopamine receptors. And you'll have subtype 1, subtype 2. Kind of getting a trend here. There's different subtypes to each of these. Now at the very end, I'll give you the high yield stuff, and I'll give you one association. M1 receptors are found in a very limited location. However, M2 and M3 are going to have a wider distribution. They'll be located in a lot of places. However, I'm going to give you one high yield location. That's it. So that way, hopefully you remember that one high yield, and at least you know something instead of knowing nothing. There's histamine receptors. There's going to be an H1 and an H2. And then finally, we've got vasopressin, also known as antidiuretic hormone, also known as angiovasopressin, all the same thing. We're going to have different receptors. You're going to have like a V1 receptor, a V2 receptor. Those are the main ones. So again, these aren't all the receptor types. These are the main receptor types. So don't be hounding me with, oh, well, what about M5? Well, who cares about M5 is all I can say. And I'll say that to you again if you ask me. So let's see. M1 receptors. An M1 receptor is going to be GQ. So it's going to be GQ, meaning it's going to activate this phospholipase C. It's going to go down through the IP3-DAG pathway. It's going to act in a GQ fashion. Now from here on out, there's going to be three options. There's going to be GS, GI, and GQ. So GS, GI, GQ. I'm just going to write this as Q, just going to write this as I, just going to write this as S, just so you know, just for reference. So M1, like I said, was GQ, it's going to be a Q. M2 is going to be I, and this is going to be an S. Oh, my apologies. You're probably thinking, what is he doing? Teaching you wrong already. Q, my apology. QIQ, okay? Now, I'm just going to fill in the blanks here pretty quickly, but I did promise you I'll give you an easy way to remember most of these now. So you can thank me later. You're probably hating me right now because I'm just going through this. I'm just giving you stuff to route memorize, but I will give you a way to remember this. GQ, this is GS, and we'll bring it home with a Q and an S. Okay, a lot of stuff here. So, as I promised, get rid of some of this down here because I think you get the picture. Okay, so first one, how do we remember this? M1, 2, and 3 QIQ. Well, you're going to kick yourself if you're a med student. Medical school sucks. That's plain and simple. Medical school sucks. If you're in medical school, you will kick yourself if you're in medical school. You're going to say to yourself, why did I do this to myself? You're going to kick yourself if you are in medical school. Now, why do I say medical school? Because first-year medical students are called M1 students. Second-year students are called M2s. Third years are M3s, and fourth years are M4s. So if you're a first-year medical student, if you're a second-year medical student, third-year medical student, you're going to kick yourself. QIQ, kick. Kind of sounds like QIQ, kick. You're going to want to kick yourself. So we just knocked off muscarinic. Boom. Adrenergics. Can we cover adrenergics? Well, I can give you half. And why do I say that? There's no, you might be thinking, well, that's bullshit. Well, it kind of is. Remember, beta, BS. That's BS. I think YouTube is probably going to censor me. It's BS. Now, why do I say BS? The betas are both S. And you're saying, well, why didn't you give me everything? That's bull blank. Well, BS. BS. It's BS that I didn't give you at all. So BS. Okay, so we knocked out two of these. Check that off. Now, if someone comes up to you, asks for some dope, be like, hey man, you got some dope? You'd be like, yeah, I got some dope. C in Spanish, S-I. C in Spanish means yes. If anybody asks you for some dope, you always tell them, yeah, I got some dope. C. You want to see it? Yeah, I got some dope. So if they ask you for dope, you're going to say C, meaning SI. Okay, we can knock off another one, as I promised. Oops, here we go. Check off this one. Okay, I can tell you vasopressin. Vasopressin. How are you going to remember vasopressin? Well, have you ever heard of Mount Vesuvius? Mount Vesuvius. It's in Naples, Italy. Let's see, Mount Vesuvius. I believe that's how it's spelled. So it's a volcano. Mount Vesuvius is a volcano in Naples, Italy. Instead of Mount Vesuvius, I'm going to tell you it's Mount Vesuvius. Mount Vesuvius. Now, why do I say that? VQVS. Mount Vesuvius. So that'll hopefully get you another one. Instead of Mount Vesuvius, it's Mount Vesuvius. VQVS. Vesuvius. Alright, and then lastly, we'll try and bring some histamine. This one's a little bit of a stretch, but for histamine, the first two letters of histamine are going to be HI. Think Hicks. Hicks from the south, yeah, they hillbillies, those Hicks. Those Hicks watch some NASCAR, those Hicks. So for histamine, you're thinking QS. So QS. So hopefully this is helping a little. Even if I can help you remember any of them, good. Just I didn't do the alphas because I don't have a good alpha way to remember it. So if you have a good one, be sure to post it in the comments section because it racked my brain and I cannot think of a good one. But hopefully you kick yourself if you're a medical student. It's BS that I didn't give you all of them. If anybody asks you for some dope, what do you say? You say C, like in Spanish. Spanish for yes. Histamine, you think Hicks. And then lastly, Vesuvius, no, Vesuvius. Okay, so now we finish up with functions. Functions. Last topic, and then we are done for the day. If you have any other recommendations for me, let me know. But we're gonna finish up with functions here. I will give you one function. Why am I just gonna give you one? Because it'll be a high yield. Now there are gonna be hundreds, thousands, millions, billions of functions. Probably not billions of functions, but there will be multiple functions. I'm just going to give you one per. So when we're going to talk about M1, I'll give you one function. When we're going to talk about A2, I'll give you one function. What about H2? I'll give you one function. One function. That's it. So if you can remember these, you'll get it 90% of the time. You'll get the main points. You'll get the basic high yield concepts. So let's go ahead and start this off. We've got alpha 1, and we've got alpha 2. I'm going to change color just to keep you on your feet. Alpha 1s, we're going to be dealing with vasculature constriction. We're going to constrict those arteries, for example. We're going to increase our blood pressure as a result. If we activate alpha 1 receptors, which will be QGQ proteins, we're going to increase our vasculature, like our artery pressure. We're going to constrict those arteries. Alright, A2, we're going to do, oops, my pen is acting up. A2, we're going to decrease insulin release from the pancreas. Decrease insulin release from the pancreas. Since I'm color coding it, I guess. Didn't know I was, but I guess I am. We've got beta 1 and beta 2. Now, these are more high yield. Alpha 1, high yield. Beta 1, high yield. Alpha 2 is good to know. If you're thinking this is way too much stuff to remember, you can forget this. You can forget alpha 2. So now let's talk about beta 1. Make sure I'm right color. Yep. This is going to increase your heart rate. Increase your heart rate. Increase your heart contractions. Heart rate. Think heart rate. High yield. Beta 2. When you see beta 2, you want to think bronchodilation. Bronchodilation? Oh, that makes sense. Yes, it's because albuterol. Albuterol is a beta 2 agonist. alpha 2 or beta 2 agonist, albuterol. It causes bronchodilation, so if someone's having an asthma attack, you'll want to dilate. Okay, so we'll continue on. We've got M1, M2, M3. Remember, you want to kick yourself QIQ if you're in medical school. Okay, so M1 is pretty much going to increase GI secretion. M2, you're going to think decrease heart rate. It'll be the opposite of beta 1. So I'm just going to draw a little line there. And then also for M3, remember a lot of functions, however, we're going to think parasympathetic. Just think parasympathetic picture. Let's see, get color coded. We're going to talk about histamine. We can talk about dopamine. And I ran out of room, so up here let's talk about vasopressin receptors. Okay, so let's talk about histamine. Histamine 1 receptors. When you think about histamine, what if I just told you histamine right now, you're thinking mast cell degranulation, you're thinking type 1 hypersensitivity possibly, you're thinking allergies. Histamine type 1 is what you would typically associate with allergies, so nose, so airway. Okay, H2, you're going to think increase stomach acid. H2, stomach acid. Dopamine receptors, remember we already covered D1 slightly in the picture of the body, but again, it's going to relax kidney, blood vessel, smooth muscle. Not that important. D2 receptors, a little more important. That's everything in the brain. So if you're thinking dopamine, you're probably going to be thinking a D2 receptor, and that's probably going to be in the brain. Now we've got 2 more to go. Home stretch. Vasopressin. 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 Vaso, vasculature. Pressin, constrict. To press it close, like press it closed. Constrict. So what it's going to do is it's going to constrict your blood vessels. Angiovasopressin. Angiovasopressin. The name says it all. Angio for like blood vessel. Pressin. It's going to constrict it. So we're going to constrict blood vessels. And then for V2 receptors, I have a video on this as well. I want you to think aquaporins. Aquaporins are going to be in your kidney. They're going to allow you to resorb water. Okay, so again, not all of them. However, I gave you high yield. Alright, if you have any questions, be sure to ask. I always appreciate comments, always appreciate likes, and if you want more great videos just like this, be sure to subscribe. Thank you very much, YouTube. It was a fun journey. I'm glad we got through this together. Until next time, peace out.